Hello dear students, in this video we're going to talk about our new activity about mitosis and meiosis. We're going to study the different events during mitosis and during meiosis, the two division, the reductional and the equational division. So let's start. So in this part we're going to talk about the basic mechanisms of sexual reproduction, documentary, meiosis, and also we're going to talk about mitosis. So let's start first with a good information here. Biology is the only science where multiplication and division, they have the same meaning. Multiplication means that they increase the number of cells. Division also, it means the increase in the number of cells. So the only science where multiplication and division, they have the same meaning is biology. So what is mitosis? Mitosis is the process by which the somatic cells, so the somatic cells diploid to N of the body, they reproduce. They produce new daughter cells. These daughter cells, they are identical. They are very similar to the mother cell, and they conserve the genetic information from one generation to another generation. So mitosis is the process by which somatic cells, diploid cells to N, they undergo division to produce new daughter cells. These daughter cells, they are identical to the mother cell, conserving and having the same genetic information. Why would the body cells need to reproduce? First, they need to increase the cellular number and to allow the growth of the individual. Example, the case of the zygote development into an embryo. Second, cause to renew the body tissues and replace the old or dead or the injured cells by new cells. Is the cell always reproducing? The cell over its lifespan will go through successive life cycles. So first, let's say, what is the life cycle? The life cycle is the set of events that the cell will undergo in order to grow and divide properly. It may be divided into interphase. Interphase, it has three checkpoints: G1, gap 1, S, synthesis phase, and G2, which is the gap 2. And we have a mitosis, which includes prophase, metaphase, anaphase, and telophase. Prophase, and they should be in order. So prophase, P, metaphase, anaphase, telophase. So we can write it as PMAT. Prophase, metaphase, prophase P, metaphase, anaphase, and telophase. This is the mammalian life cycle. It includes the interphase, G1, it's called gap 1. We have S phase, DNA synthesis, where duplication of DNA occurs in the S phase, the DNA synthesis. G2, gap 2, that prepare to the process of mitosis. Mitosis, we have, as we said, prophase, metaphase, anaphase, telophase, and finally we have the division of the cell into two daughter cells, it's called cytokinesis. So this is the mammalian cell cycle. It includes, we have the gap 1, first gap, cell is doing its everyday job. Second, we have the duplication of the DNA. So DNA is replicated or duplicated. So second gap, it prepares itself to mitosis. As you see here, the chromosomes from G1 to G2, they are of one strand of DNA, while after duplication of DNA during the S phase, chromosome, they have two strands of DNA. So this means we have duplication of DNA in the S phase. And then we move to mitosis, prophase, metaphase, anaphase, and telophase, which include the cytokinesis, where we have two daughter cells, and these daughter cells, they are identical to the parent cell or to the mother cell. Cell, at the end of the interphase, we have the chromatin. The chromatin is clear, we don't have chromosomes. Prophase, when we move to the prophase, the nuclear membrane disintegrates, the membrane around the nucleus disintegrate. We have myotatic spindle fiber, they start to form. We have the centrosome, it changes into asters and moves to the opposite poles of the cell. Chromatin condense, so that we have individual distinguished chromosomes, and each chromosome is made up of 2 chromatids. So steps of the process, this is the centromere, and we have this meiotic spindle fiber. Nuclear membrane, this is the different phases or the different events during mitosis. Nuclear membrane disintegrates, meiotic spindle fibers are formed, the centrosomes, they change into asters and they move to the opposite poles. Chromatin condense, so that individual chromosomes, they are now distinguished, and each chromosome is made up of 2 chromatids each. Phase, we have metaphase. As you see here, the metaphase, the chromosome, they line at the middle of the cell, forming what is known by equatorial plates, or you can say the chromosomes, they align at the middle of the cell, forming what's known by equatorial plates. So here, chromosomes, they line up in the middle of the cell, at the equator of the cell, forming what's known by equatorial plate. After metaphase, what do we have? Prophase, metaphase, and then we have anaphase. Anaphase, here the spindle fiber will pull the sister chromatids. So the sister chromatids are pulled to the opposite pole of the cell by the help of the spindle fiber. Each chromatid will move to the opposite pole. Here, each chromatid will move to the opposite pole of the cell. Each chromatid move to the opposite pole of the cell, forming what's known by polar ascension. So this is called a polar ascension. This is the key word here. As in metaphase, we have the equatorial plate. So the result will be two groups of identical groups of chromosomes. Each chromosome is made up of 1 chromatid at the opposite pole of the cell. So the events of anaphase, spindle fibers, they pull the sister chromatids of each chromosome apart. Each chromatid moves to the opposite pole of the cell. This is called polar ascension. And by late anaphase, the result will be 2 identical groups of chromosome, each made up of 1 chromatid at the opposite pole of the cells. Last phase, prophase, metaphase, anaphase, and now we have telophase and the cytokinesis. The telophase, we have an important note here, or the key here, it's the cleavage furrow. So this is the cleavage furrow, where we have the cell will divide into 2 cells. So the chromosomes, they form 2 identical groups at the opposite cell. These chromosomes will decondense back to the chromatin form. They will decondense to the chromatin. Nuclear membrane will reform again, will appear again around each set of the chromosome. Here the nuclear membrane will start to reappear, and we don't have spindle fiber, they disintegrate, and we have the cell membrane will constrict. This is the constriction site. The constriction site here, they will constrict, forming what's known by the cleavage furrow to divide the mother cell into 2 identical daughter cells. So the events of telophase, the chromosomes will form 2 identical groups at the opposite poles of the cell. These chromosomes will decondense back into the chromatin form. The nuclear membranes reform around each set, so nuclear membranes will reappear again. The spindle fibers disintegrate, and the asters return to the centrosome. So asters will go back to the centrosome state. Cell membrane constricts, forming a cleavage furrow to divide mother cell into 2 identical daughter cells. So here we talk about telophase. So what about the cytokinesis? Cytokinesis is the process when the cytoplasmic membrane starts to constrict, cleaving the cell into 2 identical daughter cells. We call it cytokinesis. Okay? Note too, at any stage during mitosis, the cell has 2 N chromosomes. The 2 daughter cells obtained after each cell cycle are identical to each other, as well as they are identical to the mother cell, on which both are diploid, 2N, and they contain the same genetic information. Okay? Let's move now to this figure. This figure showing the different phases of mitosis. Here, G2 of interphase, and then we have this is prophase, centrosomes. We have spindle fiber that start to form. We have chromatin condense, so each chromosome is made up of 2 chromatids each. This is the pro-metaphase, or we call it early metaphase, where the chromosomes start to move to the center of the cell, forming what's known by the equatorial plate. You have to know that in the plant cells, we don't have asterisk. So we don't have asterisk in the plant cell. This will be a difference between the plant cell and the animal cell. Metaphase, the chromosome, they line up in the middle of the cell, forming what's known by equatorial plate. Anaphase, sister chromatids will be pulled to the opposite pole of the cell, forming what's known by polar extension. On the last stage, we have telophase and cytokinesis, where the cell will divide into 2 daughter cells. These daughter cells, they are identical to the mother cell, having the same genetic information. Here is again the figure showing the different phases of mitosis. Also, this is interphase, prophase. This chromosome, they start to condense, they line up in the middle of the cell. So whenever you see a chromosome lining up in the middle of the cell, immediately this is metaphase. Chromatids moving to opposite pole of the cell, this is called anaphase. Telophase, we have 2 daughter cells that are dividing. Okay, so here are figures of the process of mitosis under the microscope. These are the prophase, metaphase. Here we have lining in the middle of the cell, forming what is known by equatorial plate. Here they move to the opposite poles, polar ascension, and here we have the cells. In this application, the following the following table shows the variation of the amount of DNA per cell in AU in a cell as a function of time in hour. So as you see here, the quantity of DNA at time 0, it was 4 AU. At time 1, it remains 1 AU. 6, it increases to 3, it increases to 6. At time 4 hours, it increases to 8 and remains constant till the 7th hour, and then it decreases back to 4. So as time passes from 1 to 4 hour, from 0 to 1 hour, the quantity of DNA per cell remains constant at 4 AU. Then as time increases from 1 to 4, the quantity of DNA increases from 4 to AU and it remains constant until the 7th hour, where it decreases to 4 AU and remains constant till the 4th hour. So the life cycle here, it lasts from 0 to the 7th hour. So the duration of the cell cycle is 7 hours. All of you know that duration of the cell cycle is duration of interphase plus duration of mitosis. So here, we present the data in the form of the graph, so you have to draw with the graph. Number 2, analyze this graph. Already we did the analysis. Indicate the duration, it will be 7 hours. This 7 hours includes the interphase plus mitosis. So you have to be careful. The cell cycle is interphase plus mitosis. Knowing that mitosis is 1 hour, so the total is 7. Mitosis is 1 hour. What is the time for the interphase? 7 minus 1 is going to be 6 hours. Schematize the cell at the 2nd hour, early 7th, and the late 7th. So you have to draw the chromosomes at these type. Schematize the chromosome at each of these instances, also 1 chromatid or 2 chromatids. What happens during interphase? You have to know that during interphase we have duplication or replication of the quantity of DNA per cell. This is the analysis. At time equals 0 hours, the quantity of DNA per cell is 4 AU. So as time passes from 0 to the 1st hour, the quantity of DNA per cell remains constant at 4 AU. Then, as time passes from 1 to 4 hours, the quantity of DNA per cell increases from 4 to 8 AU and remains constant at 8 AU. Until the 7th hour, then at the 7th hour, the quantity of DNA drops from 8 to 4 and remains constant at 4 till the 10th. So life cycle, as I said, it starts from 0 to 7, so duration is 7. Knowing that mitosis is 1 hour, so the interphase is going to be 7 minus 1, so it's 6 hours. Early 2nd hour is the interphase, early 7th is anaphase, and the late will be the interphase. You have to draw the figures that we already drawn before. 7. During interphase, the chromosomes they duplicate, allowing the duplication of DNA and duplicating the quantity of DNA per cell. As you note in plant cells, as I said before, no asters. So there's a difference between mitosis in plants and in animals. And plants there is no asters. Also, no constriction occurs at the end of telophase due to the rigid cellulose wall. So new cellulose wall will be formed, leading to two daughter cell at the end of interphase, as we already said. Okay, let's move to gametes, specialized cells with 23 chromosomes, activity 1, pages 170 and 171. Let's move now to meiosis. Mitosis is the process by which the somatic cells. Here in the meiosis, we have the mother cell of the gametes, diploid 2N, produce the gametes haploid 1N. So before meiosis begins, the mother cell of gametes go through an interphase in preparation for meiosis. Meiosis includes two divisions. We have Meiosis I and Meiosis II. Meiosis I is called reductional division, and we will see why it's called reductional. And Meiosis II is called equational division, and also we will see why it's called equational. Prophase I. Here we have something called tetrads. So we don't have the chromosomes. So we have something called crossing will occur. So this crossing over is exclusive and only in the meiosis process. During prophase I, the nuclear membrane disintegrate, like the case of prophase. Spindle fibers start to form. Centrosomes start to change into astrum. Chromatin condense, so that chromosomes are now individually distinguished, and each chromosome is made up of two chromatids. All of them, they are similar to the process of mitosis. All of them. Till now. The only difference is here in this stage. Homologous chromosomes, they pair up, they synapse, forming tetrads. They move one over the other, the chromosomes. This increases the chance of something called crossing over between the non-sister chromatids of paired homologous chromosomes. So this is the only difference. We have the tetrads. So this is the metaphase 1 of meiosis I. Okay, the formation of the tetrads. Let's move now. Metaphase 1. What happens during the metaphase 1? We don't have chromosomes, they line up. We have tetrads. They line up at the equatorial plate of the spindle fiber, at the equator of the cell. Let's move now to Anaphase 1. Anaphase 1, spindle fiber will pull, not the sister chromosomes, they will pull the homologous chromosomes. So they will pull the homologous chromosomes of each tetrad. Okay? Each chromosome will move to opposite pole of the cell, forming what's known by polar ascension, and here at the site of the crossing over. Late anaphase, we will have two groups. Each is made of two chromatids. Chromosome is made up of two chromatids, and then they move to the opposite cell. So the events of anaphase 1, spindle fibers, they pull the homologous chromosomes. In the anaphase of mitosis, the sister chromatids, if you remember. So here, pull the homologous chromosomes of each tetrad apart. Each chromosome move to the opposite pole of the cell. Polar ascension. By late anaphase, the result will be two groups, each made of two chromatids at each pole of the cell. Telophase 1. What will happen in telophase 1? During telophase 1, cell membrane constrict, forming cleavage furrow. It's trivial here. Two daughter cells obtained, where each carry one copy of chromosomes, each of two chromatids each. Obtained daughter cells, they are haploid. Number of chromosomes is reduced here. That's why meiosis I is called reductional division. So meiosis I is called reduction because the number of chromosomes is reduced. This is why it's called reductional. Daughter cells, they are different from each other, and they are different from the mother cell. The chromosome form two groups on the opposite pole of the cell. The spindle fiber disintegrate, and the asters go back to the centrosome state. So the nuclear membrane will form again. There is an important note here that the meiosis I and meiosis II, we don't have an interphase between them. Okay, so there is no meiosis, no interface between meiosis I and meiosis II. Meiosis I, we have prophase I, metaphase I, anaphase I, telophase I. Then we move prophase II, anaphase II, metaphase II, anaphase II, telophase II. Okay, guys? So, and prophase, what will happen? The mitotic spindle fiber starts to form. The centrosome, they change into asters, and the nuclear membrane disintegrate. This is very similar to that of mitosis. Very similar. Metaphase II. What will happen at metaphase II? The chromosome, they line up at the middle of the cell, forming what's known by equatorial plate. This is identical. Meiosis II, as you see till now, in prophase II and metaphase II, they are 100% identical to mitosis. But we have here two daughter cells. Let's move to anaphase II. So as I said, metaphase II, chromosome line up at the equator of the cell, forming what's known by equatorial. Anaphase II, as you see here, the mitotic spindle fiber will pull the sister chromatids of each chromosome to the opposite pole of the cell, forming what's known by polar extension. The result will be at the end two groups. Each group is made up of one chromatid, each at the opposite pole of the cell. As you see here, till now, this is identical to mitosis. Anaphase II, the spindle fibers will pull the sister chromatids apart, forming what's known by polar extension. Late anaphase, the result will be two groups of chromosome, each made up of one chromatid each at the opposite poles of the cell. Telophase II and cytokinesis. The chromosomes form two groups at the opposite pole of the cell. Here we have 1, 2, 3, 4. So these chromosomes will decondense back into what? Into the chromatin. Nuclear membrane start to form around each side, and the spindle fiber disintegrate, and the asters go back to the centrosome state. So the result here, we have 4 daughter cells, and this is an important difference between mitosis and meiosis. We have 4 daughter cells. So the chromosomes, they form two groups at the opposite pole of the cell. These chromosomes will decondense back into the chromatin, form nuclear membranes. This start to form around each set of chromosomes, and the spindle fiber disintegrate, and the asters return to the centrosome state. Here the figure showing interphase. Meiosis I separates homologous chromosomes. Meiosis I, prophase I, metaphase I, anaphase II. These are the tetrads that I told you about. This is the tetrads. Okay, these are the tetrads. Here 2n equals 6. Okay, so number of possible orientations here is 2 to the power of the number of chromosome pair. How many pairs we have? 3. So 2 to the power 3 is going to equal to 8. Comparison of mitosis and meiosis in haploid cell. This slide is going to be divided into 2. Okay, this figure. So mitosis, we have interphase, prophase, the chromosome line. The chromosomes will be 2 chromatids each, and the chromosome will line at the middle of the cell, forming equatorial plate. But in meiosis, we have prophase I, prophase, metaphase I. Here the tetrads, pairing of homologous chromosomes, as I said, is called tetrads. So tetrads, they line at the middle of the cell, homologous pair of duplicated chromosome. Instead of this, you can say tetrads, they line up at the line of the equatorial line or the equatorial plate. Here metaphase. Let's now move to the anaphase and the telophase. Anaphase and the mitosis, the sister chromatids, they are pulled apart, and we have at the end 2 daughter cells, and this is the anaphase and telophase. While for the meiosis, we have cell division 1, it's continued, and we have 2 daughter cells at the end, and then we have metaphase II, anaphase II, telophase II, and we have 2 daughter cells. So here we have 2, here we have 2, so the total will be 4 daughter cells, while in mitosis we have only 2 daughter cells. Okay, guys? So also a figure showing the differences between the mitosis and meiosis. Okay, meiosis I here for sure. And this is meiosis I and meiosis II, leading to the formation of 4 daughter cells. These 4 daughter cells, they contain 1 chromosome each, and these cells will become what's known by the gametes. So this will be the gamete. Also, this mitosis, DNA replicates 2 daughter cells, and they are identical to the mother cell. While in the meiosis, they are not identical to the mother cell, and we have 4 daughter cells. Each daughter cell will give 2. So after meiosis 1, we have 2. After meiosis 2, we're gonna have 4 daughter cells. Let's move to this very important table showing the comparison between mitosis and meiosis. So mitosis would always get place at the somatic cell. It will occur only in the somatic, while meiosis in the gonads. So, okay, sperm in the ovum aims for the growth or the renewal and replacement of the cells. For meiosis, it's production of haploid 1N gametes. One division only, producing 2 daughter cells in mitosis, so we have only 2 daughter cells, while 2 successive division in meiosis. What the meaning of this? Meiosis 1 and meiosis 2, producing 4 gametes, 4 daughter cells. Mother cell 2N and daughter cell is 2N, the same number of chromosomes per cell, while number of chromosomes is reduced into half, going from the mother cell, which is 2N, to the daughter cells or the produced gametes, which are haploid 1N. Mitosis occurs to somatic cell in all body organs, only occurs to mother cells of gametes in the gonads. Meiosis, it's made up of 1 division preceded by interphase, while meiosis 2 divisions preceded by interphase. No pairing of homologous chromosome. Here we have pairing, which is the tetrads. Daughter cells obtain the R2N, 1 chromatid per chromosome. So after meiosis 1, we have daughter cells 1N, 2 chromatids. After meiosis 2, 1N, 1 chromatid per chromosome. Note that meiosis 2, as I said, Prophase II, Metaphase II, Anaphase II, and Telophase II, they are very, very similar to that of the process of mitosis. Hope you guys understand everything, and hope that this video will help you to understand well the differences between mitosis and meiosis. Thank you guys. See you soon. Bye bye.